Welcome to one of the past HSC exam question videos. This question comes from the 2008 exam and it's question 19 and it's worth 4 marks. While I'll do in a second, I'll read the actual question. Once I read the question, give about 5 seconds to pause the video. Once you pause the video, attempt the question and then press play when you're ready. The question says, a new product has been developed to kill pathogens in drinking water. Design an experiment to test the effectiveness of the product. So when you're ready, pause the video and attempt the question. Welcome back. Alright, so this question says design, which means obviously you need to come up with your own experimental design. But at the same time, we've actually covered this in the class, or someone covered this in class, which covers something quite similar. But we need to design an experiment to test the effectiveness of the product. So remember what the product is meant to be doing. The product is meant to kill pathogens in drinking water. So the actual marking guidelines says to get full marks, so 4 out of 4 marks, you need to be able to have a appropriate data collection methods. You need to have some kind of way to collect your method, right? To collect the method, and also you need to include repetition in your experimental design. You need to be able to have at least 2 variables that are kept constant, so 2 controlled variables, and you also should have control in your experiment. And obviously, all of this is on top of just your overall experimental design. You're meant to design an experiment, and you have these things included. If you have them all 4 included, you get full marks. If any of the 3, you get 3. If any of the 2 of the 4, you get 2. And only one of those 4, you get 1. Right? So you talk about independent variable. Remember, the independent variable is what you change, and the dependent variable is what you measure. So in this case, you're going to have one set that will have, for example, lots of the product. So we have the control will have none of the actual product that kills, whereas the other one will have lots of that product that will kill, right? So the actual amount of the product is what you change. So that's your product, the amount of product is what you change, whereas what you measure would be the colony numbers, right? So the more colonies, the less effective it is. So when you measure the number of colonies, what you change is the actual amount of product you use. And the controlled variable would be, for example, your location where you get the samples from. You want to keep them the same. You want to get them from the same spot. I'll go over that in a second. You want to do things like take the same amount, same volume. You want to keep the temperature the same when you have it in the incubator, and all that kind of stuff. So these would be examples. The experiment itself that you should have done would have been the microbe experiment. Remember, there was the water sample that was part of that microbe experiment where we tested water samples and used the agar plate. So you should have had an agar plate experiment to get your marks. Right, but we'll go over this now. So these are repetition, include your controlled variables, talk about independent dependent variables because that's part of normal experimental design, and also include appropriate ways we can collect data. Right, so this is more or less what you should have done in picture form, or something similar. It doesn't have to be the same, but something similar. Right, so first you should have taken samples. In my case, I actually have 3 different things that I'm testing, but if you only chose 2 things, a control and something that has a product in it, that would be okay. So 2 to 3 samples, right? Take them from one location, so that's the same location in any given area. Then what you do is you quickly put something that pathogens can't get into, so you seal it, right? And what you also should be doing is should be labeling. So cover and label to make sure no pathogens come from the outside, and so you know what's going to be tested with these different ones. Then you bring it back to the lab, and once you're in the lab, the control one you add nothing to, right? The actual product one, you're going to add your 5 ml of the product that's meant to be killing. And I've chosen a third one. This was optional. You didn't have to choose this one, but I chose chlorine as well. So chlorine is the usual way that we sort of disinfect water. So I'm going to compare control, product, and chlorine and see which one is the most effective, right? So I add these products to it, and once I've added the products to it, what I'll do next, I'll let it wait for a bit, and then I will grab a inoculating tube, right? And obviously use good technique, 45-degree angle, making sure you don't cough into it, all that kind of stuff to make sure you don't cross-contaminate. But using good technique, I make sure that I put the inoculating tube in, do the whole S shape on an agar plate, and I use more than one, so I might do 4 or 5 agar plates per sample, so it might be 15 agar plates all up. And then once I've done all that with correct technique, I will put it into the incubator, right? Which is where temperature is set at 37 degrees Celsius. This is what's going to be kept constant again for all of them. And then after maybe 7 days, after a period of time, I would have checked the actual growth of the actual pathogens. And if, for example, the control has the most, the chlorine has a bit less, but still more than the product, that would show that the product is most effective because there's no growth at all because it killed all the pathogens, whereas the control would have had the most because nothing happened at all, whereas chlorine would have had maybe some dead, but maybe some still alive. If this was the actual result, then the product would have been the most effective. If the product and the actual control have the same amount, that would mean that it's not effective. Right, so this is what you should have done more or less, and this is what I wrote. So to test the effectiveness of the product, we have to compare how many pathogens have grown on the agar plate after 7 days. And that's meant to be the independent variable. Actually, it's meant to be, this is the dependent variable. So that's what we measure, is how many pathogens have grown, the amount of colonies of pathogens. So that's what we're trying to measure. So we're trying to measure that when using the new product, so the one that's meant to be so effective, and a control, and the traditional chlorine methods. And again, I wrote independent variable, but it's meant to be the independent variable, right? So that's the first step. We would have used these different, we would have gotten these samples, new product, control, and traditional chlorine methods. And what we want to do is we want to see how much of each of these will grow on an agar plate after 7 days to test the effectiveness of these different chlorine versus product versus nothing, which is the control. Alright, so what we did next is to measure this, we take 3 samples. This is the actual procedure, around 500 mils each. Again, the each means we keep it the same, right? Constant. We're meant to name 2 things that we keep constant from one location. The exact location should be kept the same. So we take that from the same spot and the same volume. There are 2 things we kept constant. The samples are immediately covered and labeled with control, product, and chlorine, respectively, and taken back to lab. So this is this part here, right? We've covered them, we've labeled them, and taken them back to the lab after taking them from the same sample, the same location, right? Next part would have been this part here, these 2 steps actually, right? Then prepare 5 agar plates for every sample. So 5 times 3 because we've got 3 samples. This is where the repetition comes into play, or we meant to talk about repetition. So then prepare 5 agar plates for every sample and add the same sample water into the agar plates with the help of an inoculating tube. Open the agar plates at a 45-degree angle and avoid breathing into the agar plates to avoid cross-contamination. Then place all 15 agar plates into an incubator set at 37 degrees Celsius for 17 days. One thing I actually forgot to mention is, whilst all of this would be just the correct procedure we should be using, I just forgot to say that we should be adding, before we do all this, before we add the different types of samples onto the agar plate, we should be adding 5 mils into the product, so 5 mils of the product into the product sample, and 5 mils of chlorine into the chlorine sample, just to make sure there's actually some of it inside. I just forgot to mention, I just realized I forgot to mention that, but you would have also mentioned that quickly as well. This is just the experimental design, how the experiment is designed. And then, so this is, now we have it in here, so it's in the incubator for 7 days, and the last stage was again checking how much has actually come out of the whole experiment. So now, because it says we need to have some way that we collect our data, and this is the whole noting down the color and amount of colonies that have grown over the 7-day period. If the product plate has less microbial growth than both the control and the chlorine samples, then it can be considered to be very effective, right? So if the product plate has less than both of these, that means that it actually did its job. It killed the actual pathogens better than the other 2 did. If it has more growth than the chlorine plate, but less growth than the control, then it's effective, but not as effective as our current methods, right? So if it has more, so if the product has less than the control, that's good, but more than the chlorine, that means whilst it's better than nothing, it's not as good as the current methods we have, so maybe we shouldn't be using it if we already have something which is better anyway, right? So that would have just been some statements you could have made about the outcome, the conclusion, right? And that would have basically been your experiment, right? You would have had your repetition, you've done it 5 times for every one, you would have named 2 things that would keep the same at least. You would have, in this case, the controlled ones were volume or temperature in the incubator or the place where we got it from. I just mentioned them. You can also mention safety obviously as well. Safety could be, for example, when you are dealing with the inoculating tube to make sure when you use a Bunsen burner, you don't burn yourself. That could be a safety concern. And also, there could be microbes that you grow that could be harmful if they get spread. So make sure you don't open the actual agar plates once you've used them. These could be safety concerns. You can add them in as well, because that's always good. But they come from these 2 dot points. So students will gather, process, and analyze information from secondary sources to describe ways in which drinking water can be treated and use available evidence to explain how these methods reduce the risk of infection. So here we talked about, for example, that boiling water and using chlorine can kill, which is why I added that chlorine one as well. It wasn't a requirement. If you only had 2, if you only had product versus control, that would have been okay as well. I just added that one just for show. And then this one says, this is the interesting one because it says identify the plan and choose equipment or resources to perform a first investigation to identify microbes in water or food, right? So you're asked to design an experiment that tested this. This is where this actual question came from. But because there was plan in the syllabus dot point, it means it can ask you to actually design an experiment. If it doesn't have plan, sometimes it just says perform or choose equipment and perform a first investigation. That means there won't be any design questions. But if there is a dot point which has a plan in as well, that means you can be asked to actually plan it yourself from scratch, right? I hope so. Check all the dot points and see anything that has plan. You should be expecting maybe a question that asks you to design the experiment. I hope that was useful.